We're going to look at the organization of the body's defenses in this lecture. The immune system has two branches. There's the nonspecific defenses or innate immunity and the specific immune responses or adaptive immunity. This is really important to know, so make sure you star this. The nonspecific defenses we're going to talk about in this lecture, they are nonspecific because they go after anything foreign in the body. So they don't care what it is, they will go after it. The nonspecific defenses also react fast and with the same speed and strength each time. And they don't have any memory, so they're not going to remember that, oh, I saw this cold virus a year ago. They have no memory of that. The reason that the nonspecific defenses are also called innate immunity, innate just means you're born with it. And it's just referring to the fact that the nonspecific defenses are ready to go from day one. So you don't have to train them and they don't have to learn anything. They're going to react really quickly with the same speed and strength each time. The nonspecific defenses include physical barriers, which are your skin and mucous membranes, phagocytes, inflammation, interferons, natural killer cells, and the complement system. The specific immune responses, or adaptive immunity, this involves the B and T lymphocytes. They are specific for which pathogen they recognize. So you've got millions and millions of different B and T lymphocytes, and they each recognize only one specific pathogen. So if it's not their match, they ignore it. They don't react to it. The specific immune responses are much slower to respond the first time they're exposed to their match, and you will get sick the first time. But they do develop memory, and so they will remember how bad the chickenpox was before. And so if you're exposed to the chickenpox virus again, they're going to respond much stronger and faster, and then you don't get sick the second time. So with the B lymphocytes, we're going to look at humoral immunity, and the T lymphocytes, we look at cell-mediated immunity. And the specific immune responses are also called adaptive immunity just because the B and T lymphocytes have to learn, because the first time they're slow to respond, you get sick, but the second time they've got those memory cells and you don't get sick the second time. So the organization of the body's defenses, our first line of defense would be our physical barriers, so your skin and mucous membranes. So they're really good at keeping a lot of things out of the body and from getting inside. But if something did get in, maybe through a cut in the skin or through the mucous membranes, the second line of defense is ready and waiting. So the second line of defense is the rest of the nonspecific defenses. So that would be like your phagocytes and inflammation and natural killer cells and all kinds of stuff there. So second line of defense is just the rest of the nonspecific defenses. And then the third line of defense, this is kind of like your big guns. This would be your B and T lymphocytes. So they will come in and help destroy, whatever is inside of you and help out the rest of the nonspecific defenses. So here's just a list of the nonspecific defenses. We're going to talk about most of these as we go through. And we're going to start with the first nonspecific defense, our physical barriers, the skin and mucous membranes. So that's your first line of defense. That keeps a lot of things from getting into your body. The skin is a great physical barrier because you've got the keratin protein in the skin cells, and that makes it a nice waterproof barrier. You've got tight junctions in between all those epithelial cells. It's dry, it's salty, a lot of microorganisms can't live in that kind of environment. It's kind of like a desert. And then you've also got oil on the skin that inhibits bacterial growth as well. The mucous membranes, any place you've got openings in your body, you'll have mucous membranes. And they also extend down into like your GI tract, all that's mucous membranes as well. So the mucous membranes will make mucus, which will trap the pathogens, and we will get rid of them that way so they can't go further in the body. And then also we secrete some antimicrobial substances like enzymes called lysozymes. These are in the saliva and tears, and They will break apart the bacterial cell wall and kill bacteria, and then also the stomach acid. pH of 2, very acidic, that will also kill any bacteria that you might have ingested. And this is just showing you the physical barriers, the skin and mucous membranes, and you've got some chemicals there too, like the stomach acid, the mucus, and lysozymes and other things that will help prevent things from getting into your body. So we just did our first line of defense, the physical barriers, the skin and mucous membranes, which are non-specific. They keep out everything. But if something did get in past the skin and mucous membranes, we've got our second line of defense, the rest of our non-specific defenses. So that's what we're going to look at now. So some of the white blood cells are phagocytes, and they are non-specific. They eat everything and they will act very quickly. The 4 types of white blood cells that are phagocytes that would be important to remember are neutrophils, monocytes, macrophages, and dendritic cells. So neutrophils, monocytes, macrophages, and dendritic cells. So the phagocytes will come up and engulf these bacteria here, for example. They'll surround it with their plasma membrane and then bring that in. And then the lysosomes will fuse with the phagosome there, and the digestive enzymes will then break apart whatever it was that they took in. Our next nonspecific defense is inflammation. And inflammation could be triggered by a pathogen or by tissue damage. So for example, maybe you sprained your ankle, that would also cause inflammation. The reason we have inflammation is it helps clean up the damaged or infected area and allows you to repair it. There are 5 signs of inflammation that are important to know, and these signs of inflammation are all caused by the chemical histamine, which will be released by mast cells and basophils. The signs of inflammation are redness, swelling, heat, pain, and loss of function. There's 5 steps in inflammation. You do not have to memorize these 5 steps. Just understand what's going on during inflammation. That's all you need to know. So you don't need to memorize these. So let's say that we get a cut on your hand and there's some bacteria that have gotten in there and maybe some dirt as well. There's some macrophages that live right underneath the skin, and so they will start acting immediately and start phagocytizing and eating the bacteria and the dirt. And they're also going to release some cytokines, some chemical messengers that will call for reinforcements. And then mast cells and basophils that are in the area, they're going to secrete histamine. And histamine is going to cause 2 things. So this is important to remember. So it's going to dilate the capillaries, and it's also going to increase the permeability of the capillaries. So by dilating the capillaries, we're going to increase blood flow into that injured area. And that will cause that injured area to turn red and also feel hotter to the touch. So that'll cause the redness and heat that we see with inflammation. And the reason you want to increase the blood flow to that area is that will allow more white blood cells to get into the injured area. And then we're also going to increase the permeability of the capillaries. So we're going to make the gaps in the walls of the capillary wider, and that will allow fluid from the blood to leak out more into the tissue. And also some of the proteins will leak out. So we need things like clotting factors and complement proteins and other things. And then also the increased permeability will make it easier for the white blood cells that are in the blood to move out into the injured area. And with the increased permeability of the capillaries, with more fluid moving out into the injured area, that's going to cause the signs of inflammation, of swelling. And then if it's swollen, it'll be painful. And then if it's painful, you might not want to use the area. So that would be loss of function. And this is just a close-up view of the 2 effects of histamine. So here's before the histamine gets to the capillaries and really starts causing the changes. And you can see the histamine here is being released by mast cells, and it's also released by basophils too. So the two effects of histamine, you can see that we have vasodilation of the capillary. So we're increasing the blood flow into the area. That'll bring more white blood cells. And then the second effect of histamine is it's going to increase the permeability of the capillaries. So more of the fluid from the blood will leak out into the tissue, and also it makes it easier for the white blood cells to get out there and help clean up the mess out there. So those are the two effects of histamine. It causes vasodilation and increases the capillary permeability. And those effects of histamine cause all 5 signs of the inflammation. So mast cells and basophils are also going to be secreting some heparin, which will prevent the blood from clotting in the area at first. And that's just because you want the blood flowing and making sure that your white blood cells can get into that area. And clotting factors will leak out of the blood into the injured area because you've increased the permeability of the capillaries, and that will obviously stop the bleeding. And then we're also going to throw a clot around the microorganisms that have gotten in, or the dirt or whatever's in there. And the clot is like a big net, and that will trap the microorganisms and prevent them from escaping and getting into other areas of the body. And now finally reinforcements arrive. Neutrophils are always the first ones to arrive, and then macrophages will come in a little bit later. So you've had to make more of these in the bone marrow, and now they're finally arriving. So we need to make sure that the macrophages and neutrophils that are arriving know where the injured area is and know where to stop and leave the blood and get out into the injured area. So we've got a couple of steps here. There are some cytokines that are released, little chemical messengers that say, hey, this is the spot. So we'll have margination occur first, and this is where the white blood cells will move to the margins, the edges of the capillary. And then they're going to bind to some little protein hooks that are put out by the capillary wall cells, and the white blood cells will grab onto those, and that stops them from flowing by. And then the third step is diapedesis, and diapedesis just means to jump through. So this is talking about the white blood cells squeezing through the gaps in the capillary walls. That's diapedesis. And then once they're in the tissue, they're going to do chemotaxis, which just means they're going to follow a little chemical scent trail to find out where the injured area is and follow that trail. And then once we've got all our reinforcements there, they will then finish cleaning up the mess and phagocytizing everything, getting rid of the microorganisms, the dirt, any dead cell parts. They'll clean all that up. And once the area has been cleaned up and is free of infection, you've gotten rid of the dead cells and dirt and stuff like that, then the tissue can start to heal and you can start to form new skin cells over there. So that's why it's really important with wound care to make sure that we get rid of the infection, and we also need to make sure that the wound is cleaned out really well, because it won't start healing until that's done. Our next non-specific defense are interferons. Interferons are cytokines that are going to be secreted by a cell that's infected with a virus. And it's going to tell the surrounding cells that a virus might be coming their way and get ready, and that'll prevent the spread of the virus to other cells. So here's just a picture of the interferons. So you can see this cell on the left is infected with these blue viruses here, and this cell realizes that it's a goner and it's infected with virus, so it's going to warn its neighbors. So it secretes these cytokines, these chemical messengers called interferons, and that'll tell the neighboring cells to get ready for a possible virus coming their way. So these surrounding cells might do things like shut down their ribosomes so they're not making any proteins to make new virus. So interferons are called that because they interfere with the viruses, and prevent them from spreading to other cells. And they're also non-specific because it doesn't matter what type of virus it is, any virus will stimulate the secretion of these interferons. And our next non-specific defense are natural killer cells. Natural killer cells are one of the 3 types of lymphocytes. So remember the lymphocytes, we have B lymphocytes, T lymphocytes, and then we have natural killer cells. Natural killer cells are non-specific. They're going to kill any virus-infected cell or any cancer cell. It doesn't matter what they're infected with or what type of cancer. But the B and T lymphocytes are very specific. They have very specific pathogens that they recognize, and if it's not their match, they won't go after it. But natural killer cells are non-specific. So once they find a cell that's infected or cancerous, they will secrete some perforins, which are proteins, that will make a hole in the abnormal body cell. And then through that hole, they'll pour in some chemicals called granzymes. And granzymes will trigger that cell to do apoptosis or to commit suicide. And that's how it kills the cell. And we're going to see this method of killing again because it is a lymphocyte and it is related to the cytotoxic T lymphocytes, which are going to kill the same way as the natural killer cells. The last non-specific defense we're going to talk about is the complement system. The complement system consists of 30 different plasma proteins that are made by the liver, and they just float around in the blood in the inactive form. And then once they're activated, they could do one of 3 things. So they could destroy a cell, particularly bacteria, they're good at that. They can also cause inflammation, and they can also act as an opsonin. So opsonin, that word means to make tasty, so they end up coating the bacteria, and that makes it easier for the phagocytes to grab onto it and eat it. So that's what an opsonin does. So let's take a look at our complement proteins. Activating the complement system, we set up a cascade. So we end up activating the first complement protein, which will then activate the next, and then the next, and the next, until you get your end result, where maybe the complement proteins are forming what's called a membrane attack complex, which lyses the cell, or maybe we cause inflammation, or maybe it acts as an opsonin and coats the microorganism to make it easier for the phagocytes to eat. So activating the complement system, there's basically 2 ways to do it. There are 3 different pathways, but I'm not going to make you memorize that. So in order to activate the complement system and get that cascade going, either the complement proteins come into contact with the bacterial cell and that sets it off, or if the bacterial cell is coated with antibodies, then that'll activate the complement system as well. So either coming into contact with the bacterial cell or coming into contact with the antibody that's bound to the bacterial cell, that will also set off the complement system. And then just actions of the complement system, it can do 3 things. One, it could lyse the cell. So for example, we've triggered our complement system, we've activated the first complement protein, it bound to the bacterial cell and then activates the next, the next, the next, and then finally you end up forming as the end result with these complement proteins this membrane attack complex, or MAC. And that just forms a hole in the bacterial cell. Water will rush in and the bacterial cell will actually explode, which is pretty cool. And then obviously the bacteria is dead. Another way that the complement system can act is it can act as an opsonin. So let's take a look at that. So opsonins are just things that coat the microorganism. So maybe we've got a bunch of complement proteins that are coating this bacterial cell, and that makes it easier for the phagocyte to grab onto the bacterial cell and eat it. Because some of these microorganisms are a little bit slippery, and so if you coat them with the complement protein, that makes it easier for the phagocytes to eat them. And another thing that acts as an opsonin are antibodies, which we'll look at in another lecture. And don't worry about the acute phase proteins, I won't ask you about those.